Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at GeneCopia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I'd like to thank you all for joining us for today's webinar entitled Strategies for Effective CRISPR-Mediated Gene Modification. We really appreciate it. Before I go into the webinar, I'd just like to go over a few practical matters. First, you may ask me questions at any time. But in order to do so, you won't be able to speak vocally to me because your microphones will be muted. Instead, you'll need to type your questions in the questions box, which is in your control panel. If you can't see your control panel, it might be hidden, in which case look for a small strip of buttons on one side of the screen. One of those buttons will be orange with a white arrow on it, and when you click on that button, you'll then be able to see your control panel and hence the questions box. Secondly, in the interest of keeping the talk going smoothly, I'm going to wait until the end of the talk to answer your questions. And in that case, I will read each question as well as the answer to that question out loud for everyone to hear. And finally, I will be sending you a couple of links via the chat box, and that's also located in the control panel. So that way, that'll allow you to download various documents right in the webinar if you want to. Okay, so with that, let's go into the webinar. Okay, so here are the goals of this presentation. First, to help guide you through the basic considerations, steps, and potential problems for using CRISPR to modify genes and cell lines. Now, I emphasize cell lines because that's what most of our customers use our tools for. That's what they are. At least the off-the-shelf tools are primarily designed for. But of course, we can provide solutions for other situations like transgenic animals, but that's a little beyond the scope of today's webinar. Today is mainly about using these tools in cell lines. And so then, of course, I'm going to present the CRISPR-based tools from GeneCopia for modifying genes in cell lines. And then finally, I'd like to have a question and answer session. So it'd be great if this could be kind of like a workshop, more of a workshop than a lecture. So please bring your questions ready. Okay, so I want to now tell you who we are at GeneCopia and what we're about. We were founded back in 1999 as a company that provided large collections. In fact, they're genome-wide collections of mostly human and mouse ORF clones or open reading frame clones. These are plasmid clones that contain the open reading frames of genes, and they're ready to express in cells or in bacterial systems or various other systems the day you get them. And we've since branched out into other clone types like promoter clones, gene promoter clones, microRNA solutions, various types of microRNA-related clones, CRISPR clones, which of course is the focus of today's talk, and shRNA clone set solutions. In addition, we have viral systems for lentivirus and adeno-associated virus, or AAV. These come in either the form of, at least for lentivirus, packaging kits, where you can take lentiviral plasmids from us or from other sources and make your own virus. Or even better than that, you can purchase ready-to-use viral particles from us. And those are usable in either cell lines or in animal models. Further, we have a bunch of stable cell line solutions, primarily Cas9 stable cell lines. I'll talk a little bit about that in today's webinar, as well as labeled cancer cell lines and cancer biomarker mutant cell lines, as well as custom cell line services. In addition, we have a number of kits and reagents, such as transfection reagents. We have our own endofectin line of transfection reagents for various types of cell lines. We have luciferase kits, luciferase assay kits for either the classic Firefly luciferase system or the Ghostia luciferase system for secreted luciferases. We have FISH probes, fluorescence in situ hybridization probes, indel detection kits. I'll talk a little bit about that today, as well as a number of cloning kits. And finally, we have a large selection of fluorescent detection reagents for cell function assays, like for apoptosis. We have nucleic acid detection reagents like DNA gel stains or DNA quantitation, nucleic acid quantitation kits. cell structure probes, and fluorescent dyes. But of course, you're here to learn about CRISPR solutions, so let's talk about that. So this is a basic workflow for using CRISPR in cell lines, and it shows the steps as well as the main GeneCopia CRISPR products for doing genome editing in cell lines. We have products that can cover pretty much every step of the process, from sequencing the target region and design and construction of single guide RNA clones and HDR clones if needed, and I'll talk a little bit about that. We also have solutions for validation of your CRISPR solutions in cell lines, as well as determining copy number via FISH probes that I just mentioned. We have products for the transfection or transduction steps, either with high efficiency transfection reagents or lentiviral particles. And finally, we have a number of product solutions for the screening step, which is extremely critical. And I'll talk about that in a little bit. So this is the outline. I'm going to go over first, give you a brief introduction to CRISPR technologies and applications. Then I'm going to talk about the strategy considerations, things you need to think about before you even design your experiment, your gene editing experiment. Next, I'm going to talk about delivery methods for the gene editing CRISPR reagents. Then I'm going to talk about preparation, so how to actually get started with your stuff, with the stuff that you receive. Then I'm going to go into some of the screening steps and considerations, and then finally, I'm going to talk about things to look out for, like pitfalls and things that can possibly trip you up if you're not thinking about them carefully. But of course, you are going to think about them carefully. Okay, so let's talk about CRISPR technology. So CRISPR has been around now for genome engineering for maybe the last 5 to 6 years. It really caught on in late 2013, early 2014. Okay, and it's still going strong. It has virtually supplanted all other gene modification technologies, which include things like TALEN and zinc finger nucleases. I'm not going to talk about those, but those are some of the older technologies. And even to this day, even after all this time, still the most widely used version of CRISPR, which is found in most bacteria and all archaea as an adaptive immune mechanism in those organisms. Still, the most widely used version of CRISPR-Cas9 that's in use currently comes from Streptococcus pyogenes, or S. pyogenes, which is the same bacterium that causes strep throat. Okay, and this is a diagram that shows some of the basic features of the Strep pyogenes CRISPR-Cas9 system as it's used for gene editing. It's a three-component system. So first, there's the Cas9 nuclease, which is denoted by this yellow shape here in this figure. Okay, but this does not have nuclease activity on its own. Instead, it needs to be complexed with two RNA species. The first is shown here, not the red part, but the black and blue part, which is known as the CRISPR RNA or crRNA, and that provides target site specificity. So what happens is this contains a 17 to 20 nucleotide target sequence from the genome. Okay, and you can program this sequence in, so you can synthesize an RNA with essentially any target site in the genome you want, and then it is complexed with another species shown in red, known as the transactivating CRISPR RNA or tracer RNA. And in fact, it was shown a long time ago that these two RNA species can be fused into one single guide RNA. Okay, so when this complex comes together, it then is guided to the same target sequence in the genome if it is found upstream of an NGG PAM, which stands for proto space or adjacent motif site, that's immediately downstream. Okay, so when the when the Cas9 and single-guide RNA complex finds this PAM site and the target sequence, it then binds to it by Watson-Crick base pairing and causes a double-strand break in the genome. Okay, as shown here. This double-strand break has to be repaired or the cell will die. So there are two major mechanisms by which the double-strand breaks are repaired. One is known as NHEJ, or non-homologous end joining. That's simply sticking the two ends of the broken chromosome back together. But this is a highly error-prone process, so that can be used. And so what often happens is, and I'll talk about this in a little bit, is you get frame shift mutations which can be used to knock the gene out. And the second pathway is known as homologous recombination. That requires the presence of what's known as a donor, shown here, which is an exogenously added piece of DNA that's homologous to the target region on both sides of the break. And when you have homologous recombination, you get repair of the break using the donor DNA as a template. And so what this allows people to do is to insert sequences in between the homologous arms, and then you can get a knock-in of virtually any sequence that you like. So it can be a marker, it can be a mutation, it can be a transgene, it can be just about anything, any sequence. Okay, so that's the major version of the most widely used version of CRISPR-Cas9 today, that's used mainly for gene modification. Now, there have since been a number of technological variants that have been developed, and these are shown in this table. The top row shows what I just showed you, the strep pyogenes Cas9 or SP-Cas9, which requires, as I mentioned, an NGG PAM site. But there's also a recently developed variant called SP-Cas9NG, which actually only requires an NG PAM. This only came out a few months ago. It's fairly new, and this is nice because it allows you it gives you broader target site specificity, so you can find your target region in more places in the genome. Also, a number of variants have been developed that address some of the off-targeting issues of Cas9. I'm not really going to talk about this too much, but CRISPR-Cas9 has a propensity for off-targeting. So a number of variants, such as the Cas9HF or ESP-Cas9 have been developed, or also a brand new version called Hi-Fi Cas9. And these also have the NGG PAM requirement, but they are mutagenized high-fidelity SP-Cas9 versions, which give almost as high level of on-target efficiency as SP-Cas9, but with much lower off-targeting issues. In addition, there have been Cas9s from other species that have developed, or Cas enzymes from other species, such as CPF1, also known as Cas12a, which are nucleases from one of two different bacterium. And this has a completely different PAM requirement. And this can be used for most gene editing applications like gene knockout, knock-in, tagging, and mutagenesis. Another species of Cas9, SA-Cas9, has also been used. This comes from Staphylococcus aureus, and it's smaller than SP-Cas9, and so its main usage is with adeno-associated virus, or AAV, and that has yet a completely different PAM requirement. Now, this is not as widely used as SP-Cas9, though, in general, because it doesn't have as high efficiency or specificity as SP-Cas9. In addition, there's also the dead Cas9, which is a nuclease dead version of SP-Cas9, which is used for tethering to effectors, like things like base editors or enzymes used for not enzymes, but transcriptional activators and repressors for gene activation and repression. I'm not going to talk about this today, but that is an interesting application. And finally, but though not completely finally, this is the last major version I'm going to talk about. There's Cas13, which is an enzyme from L. Wadia that targets RNA. And this is mainly used for RNA knockdown or mutagenesis. Somewhat similar to RNAi, but You can do mutagenesis, which RNAi cannot do. Okay, so now let's talk about some of the applications for CRISPR, some of the most widely used applications. One of the most widely used applications is gene knockouts, and one way to do this is by getting frame shifts using the NHEJ pathway I mentioned before. So what happens is if you have CRISPR that finds its target site in the genome, as shown here, it will make a double-strand break 3 nucleotides upstream of the PAM site. And the repair pathway, the NHEJ repair pathway, has several possible outcomes. One is that it can stick the 2 chromosomes back together completely faithfully, and you'll get the exact target sequence back. Okay, but if that happens, that is still a substrate for Cas9, so it can cut again. But what also happens is you can get chewing back from both ends, which can lead to indel mutations. Okay, and so these indel mutations come in several different variations. So you can get a multiple of 3 plus or minus 1 indel frame shift. Okay, so that could be like, say, a single base pair insertion or deletion would be like a multiple of 3 plus or minus 1. Okay, or you can get a multiple of 3 plus or minus 2. Both of these situations will lead to a frame shift. In most cases, frame shifts will cause destabilization of the RNA and premature stop codons, and so you'll get a knockout. Although it doesn't always happen, because in some special cases you can get frame shifting by the translation machinery. You can also get an indel that's a multiple of 3. Okay, so you can get an insertion of 3 or 6 base pairs, although those are very uncommon. But you can also get deletions of 3, 6, 9, or bigger indels, and those may or may not lead to a knockout. They're in frames, so they may produce a protein, but they may not. And so again, this is a summary of what I just told you about gene knockouts by frame shifts. NHEJ mediated indels occur stochastically with unpredictable lengths. Again, we've seen indels as short as 1 base pair, and we've seen them greater than 200 base pairs. And it seems to be completely unpredictable. In general, 2/3 of the indels, since it is random, will cause a frame shift. In most cases, this will knock the gene out. Also, in general, 1/3 of the indels will be in frame. And as I mentioned, this may or may not knock the gene out, or it could also lead to the production of a protein with a new or altered function. So if you're going for a knockout, it's usually good to try to screen for alleles that are indels, because that does give you the best chance of getting a knockout. Sorry, I mean a frame shifting indel. And then typically each chromosome will contain a different indel or no indel at all. So in a diploid cell, only 4/9 of diploid cells that contain 2 indels will contain 2 frame shift indels. So these are considerations to take into account when you're doing a knockout experiment. Another way of knocking a gene out using CRISPR is to generate large deletions in genes. So we might be talking tens or even hundreds of kbs rather than tens or hundreds of base pairs. And the way people have done that is to design CRISPR guide RNAs that can simultaneously target 2 different portions of a gene, which again can be spread very far apart. And when you do this, CRISPR is great for multitasking, so you can introduce 2 of these into your cells at the same time. When you do this, you can get a deletion between this large region and then leading to a complete or nearly complete deletion of a gene, exons and introns included. And then the next thing I want to talk about is gene mutagenesis. That's another widely used application for CRISPR. One way to do this is by homology-dependent repair, also known as HDR, and this requires a donor. So if you target a CRISPR to a particular region of the genome, let's say you want to mutate a base to cause, let's say you want to, like in this example, change a T to a G, okay? And you might be doing this because Oh, I'm sorry, I just got ahead of myself here. You might be introducing this. In this case, you would be introducing a serine to alanine substitution mutation in this position. So if you target your single guide RNA CRISPR-Cas9 complex near this site you want to mutate, and then introduce at the same time an oligonucleotide donor which contains the mutation, you will then get knock-in of this mutation to the site, and then successful mutagenesis in the modified chromosome. This is a very widely used application. Another way to do gene mutagenesis is to use base editors. This is, as I mentioned before, does not cause double-strand breaks. Instead, these are cytidine or adenine deaminases that are fused to a special version of Cas9 known as Cas9 micase, which can only make one cut. And when you do this, it permits changing of the bases without double-strand breaks, and you can make any number of changes, including cytidine to thymidine, G to A to G, and T to C. So this is a way of doing mutagenesis without double-strand breaks, which is nice because double-strand breaks can be mutagenic in and of themselves. And the next application I want to talk about is gene tagging. This can be done either with oligodonors if the tag is small, like in the case of FLAG, or with large plasmid donors in the case of larger tags like GFP or RFP. This is shown here schematically, where you have CRISPR targeting near the stop codon of a particular gene, and then you introduce a homologous donor which contains the GFP just upstream of the stop codon. And then when this is successful, you get insertion of the GFP and to create your fusion protein. Another widely used application is gene knock-in. This is for transgenesis. So let's say you want to knock in a human gene in a mouse cell line, or just a marker like GFP into a cell line for tracking purposes. This is done. This has traditionally been done using lentivirus, but the major disadvantage of using lentivirus is that insertion is random, so you can get deleterious insertions that way. But another way to do that is to use the human and mouse safe harbor sites. These are particular sites in the human and mouse genomes that ensure transcription competency and where insertion prevents no known adverse effects on cells. So safe harbor integration provides low copy number and transgene, low copy number of the transgene and close to physiological levels of expression. And finally, the last application I wanted to mention, although I'm not going to go into detail about this, is gene activation or repression. I did allude to this earlier, where transcriptional effectors like repressors and activators can be fused to a nuclease dead version of Cas9. And when these are carefully targeted to usually the promoter regions of genes, these can lead to activation or repression of the gene without changing the nucleotide sequence. Okay, so now let's talk about strategy considerations, things to keep in mind before you start. Okay, so the first thing to keep in mind to think about is what type of application are you doing? It could be gene knockout, mutagenesis, transgenesis, etc. This decision is important because it can affect the efficiency and the degree of difficulty of screening for the modification. The next thing to keep in mind is what type of cell line are you using? Are you using primary cells or immortalized cells? This is critical because, as you do know, primary cells have a limited doubling time, so it is very challenging to do any kind of gene modification experiments in these cell lines. Next, can the cell line be transfected? If it does not transfect well or not at all, then you might need to use lentivirus for delivery. However, lentivirus cannot be used for HDR applications because lentiviruses enter the cells as RNA, but HDR donors must remain at all times and enter the cell as DNA. Okay. And then there are other cell line considerations such as growth rate, growth characteristics, meaning like are they inherited cells versus suspension growth cells, and target gene copy number. So these are the major questions you need to ask yourself and keep in mind as you're designing the experiment and before you start. Okay, so now let's talk about delivery methods for the reagents. Okay, so I'm going to talk about 5 different delivery methods for mainly for cell line delivery. Okay, the first method and the most traditional method is to use Cas9 on sgRNA plasmid DNA by transfection or electroporation. Okay, so this shows a typical GeneCopia Cas9 sgRNA plasmid. We call this an all-in-one plasmid because it encodes both the Cas9 nuclease under the control of a strong constitutive promoter, as well as the single guide RNA. And these plasmids can be transfected by standard methodologies using either liposome-mediated transfection reagents or electroporation. They may or may not contain a fluorescent marker, which is useful for sorting. And again, this has been a widely used, traditionally used method for gene editing, but it may not be the best method for various reasons. The Cas9 and guide RNA still need to be transcribed. Cas9 needs to be translated. And also plasmids can integrate, which is sometimes okay, but isn't always desirable. So for that, there are alternative methods for Cas9 delivery that I'd like to talk about. So the delivery method 2 is to use RNA delivery methods. So one can co-transfect in vitro transcribed Cas9 mRNA with sgRNA. And the sgRNA can be either encoded on a plasmid, in vitro transcribed, or synthetic. And the major advantage is that this is plasmid free, so it cannot integrate. But Cas9 still needs to be translated, and the Cas9 mRNA, while it does work, you do have to be careful. It's like a lot of other in vitro transcribed mRNA, it can be unstable, so you have to be very careful with its handling. That brings me to method 3, which we think is the most promising method, and that's to use Cas9 ribonucleoprotein, or RNP. This is where you preform a complex of Cas9 protein as well as sgRNA in vitro to form a ribonucleoprotein or RNP complex. Now, the sgRNA itself can be either in vitro transcribed or chemically synthesized. And this figure shown here shows actually a dCas9 sgRNA complex, but it's the same type of complex as Cas9, wild-type Cas9 with sgRNA. Now you can transfect your cells with the RNP complex using either lipid transfection reagent or electroporation. And the reason we think this is the most promising method is because the efficiency of indel formation is generally similar to, or even maybe a little bit higher than that of plasmid transfection, but it has lower off-targeting compared with that of plasmid transfection. And Cas9 protein and sgRNA does not integrate. So it has a further advantage in that you don't get unwanted integration. So this brings me to the fact that I wanted to introduce to you a product that's coming soon from GeneCopia. We're very excited. And that's our GeneHero Cas9 nuclease. That's Cas9 protein that we have produced ourselves from bacterial expression. And shown here is some preliminary data of either Cas9 protein complex with sgRNA. This shows that this leads to, this is a T7 nuclease 1 assay where a PCR product containing the target site is digested with an enzyme that detects indel formation. And so in this gel on the left-hand side, where we transfect cells with Cas9 protein and sgRNA, we see the expected bands from this digest. But with sgRNA only, we do not see the digestion bands. And so again, we're very excited about the upcoming introduction of this product. And it also works very well in transfection with our own transfection reagent, Endofectin Max. Okay, the next delivery method I wanted to talk about is lentivirus. And so I pose the question here, why lentivirus? Well, there are a lot of different situations where you might want to use lentiviral delivery instead of either plasmid or RNA or protein transfection. Number one is that DNA transfection is not always possible or practical. Some cell lines are difficult or impossible to transfect. And also, most mammalian cells support infection by engineered lentivirus, so it is a very efficient way of delivering reagents to cells. But of course, as I mentioned, lentivirus does lead to random integration. So if you don't want that, then lentivirus may not be the method for you. But lentivirus, again, is still a widely used method. And so there are several ways of getting lentivirus into cells. At GeneCopia, we have a two-component system. One is lentiviruses that express the Cas9 nuclease by itself. And shown here is the Cas9 nuclease lentiviral vector. And the second component is a lentiviral vector that expresses the single guide RNA by itself. Now, you could combine these into an all-in-one plasmid, but we generally don't recommend it because the size of the Cas9 nuclease gene is large, so it leads to reduced lentivirus titers. And so we generally recommend either co-transducing cells with both lentiviruses at the same time, or what we think is even better is that you first establish or obtain a Cas9 stable cell line first, and then infect these cells with sgRNA expressing lentivirus. And that leads me to the next GeneCopia product I wanted to introduce, which is our GeneHero Cas9 stable cell lines. These are cell lines with Cas9 already stably integrated into the genome. And so we have greater than 70 such pre-made cell lines available in human, mouse, and rat. And so these are great because, as I mentioned, we think the best situation for using lentiviral-mediated CRISPR delivery is to start with a Cas9 stable cell line, and we have 70 such cell lines already made off the shelf, ready to go, so you don't have to make them yourself. And they're in many commonly used cell lines such as HEC293, NH3T3, HCT116, etc. And these are functionally validated for Cas9 activity, so you know you're getting a high-quality product to begin with. And again, they're ideal for lentiviral CRISPR applications and also for sgRNA libraries. And if you would like to learn more about Cas9 stable cell lines for CRISPR delivery, we have a technical note that talks about this. And this is the download link, but I don't expect you to memorize or write this link down. Instead, I'm going to copy and paste it into your chat box. And remember, I mentioned the chat box in the early, the beginning of the talk. So I just sent that to you via the chat box, so you can download that if you want at your leisure, anytime during the talk. Okay, and the last method I wanted to mention for CRISPR-Cas9 delivery is to use adeno-associated virus. Now, this is not, as I mentioned, this is not widely used for cell lines mainly because this is primarily used with Staphylococcus aureus Cas9 that I mentioned before. You can put Strep pyogenes Cas9 into adenovirus, but it's right at the limit of the size limit of the adeno-associated virus genome, so it's difficult to stuff it in there and get good titers. So most people use adeno-associated virus with the SA Cas9, as it's called, which, as I mentioned before, isn't as efficient as Strep pyogenes Cas9. So most people use this for animal model work, and it's most likely going to be used mostly in gene therapy applications. But AAV is a viable option, and it can be used in cell lines. So what is AAV? Well, it's a virus that was That's was first identified as a co-isolative adenovirus, hence the name. It has a single-stranded DNA genome of 4.7 kb, and as I mentioned, SP-Cas9 is 4.3 kb, so it's right at the size limit. So it favors the use of smaller Cas9s like SA-Cas9. Engineered AAV, unlike lentivirus, does not integrate into genomic DNA, and it infects both non-dividing as well as dividing cells. But as I mentioned, it's most frequently used in vivo. Okay, so now let's talk about the preparatory steps for doing CRISPR-Cas9 applications in cell lines. Okay, so the first step we recommend is to choose your application, like I mentioned before, gene knockout, mutagenesis, etc. Again, as I mentioned earlier, as one of the considerations, it can affect the efficiency and degree of difficulty in screening for modifications. And the next step is to use GeneCopia's website to choose targets for knockout, or contact us for custom single-guide RNA and donor design if applicable. So again, I mentioned our website. This is our home page of our website. And so the first step, if let's say you want to knock out a gene and you want to order reagents from GeneCopia to knock that gene out, is to go to our website and type your gene symbol into the search bar at the top of the page. In this case, I typed in INS, which is for the human insulin gene. Okay. And then when you do this, you just press the magnifying glass, and when you will then next be presented with a search results page where you can narrow the results down by product type as well as delivery format, shown here if you scroll down a little bit. And so what I'm going to do is, I want to knock out the human insulin gene, so I'm going to filter my results by pressing the human checkbox, clone checkbox, and CRISPR checkbox. Okay, so that leaves me with a number of still a number of choices based mainly on accession number, but I happen to know the accession number of the human insulin gene, so I click on it, and this brings me to the product ID page. Okay, and so this is the product ID page as it's presented to you, and you need to scroll down to see the different product types. So the default page is for plasmid clones, but you can also select other product types. Like you can even use TALEN if you want. Again, that's an older technology, but we're talking about CRISPR. Or you can order knockout HDR donors, as well as other options like indel detection primers or lentiviral particles. But we're just going to go with clones for today, with plasmid clones. So usually we like to go for the all-in-one clones that are shown underneath here. And so we would need to scroll down and select either a set of one, or either a single clone or a set of three. But you get your product selection page here, and then you can just order them by placing them in the shopping cart, and then you'll receive within 2 weeks plasmid clones for doing CRISPR-mediated knockout. Okay, so the next step is to determine that we recommend. This is not essential, but we recommend determining the DNA sequence of the single guide RNA target site in your cell line. And the reason we do that is to ensure the highest possible nucleotide match between the single guide RNA and the genome. Okay, now that's again usually not necessary, but it might be a good step because you might have a polymorphism in your single guide RNA target sequence that can decrease or disrupt single guide RNA recognition by CRISPR. And so in so doing, if you're going to undertake this step, you can consider using our Smart Join blunt-end PCR cloning kit. So in this case, you would generate a PCR product using primers flanking the target site, and then you can clone this PCR product into an E. coli plasmid using our Smart Join blunt-end PCR cloning kit. These are the features. It provides a convenient method for high-efficiency cloning of blunt-end PCR products. Higher positive ligation rate and true positive colonies are provided by the elimination of self-ligating products. It also allows the cloning of blend-end PCR products without additional sequences, like adding adapters or adding restriction sites. You can just do the PCR using any primers that flank the site and clone them straight into your vector. And it's ideal for sequencing regions of genomic DNA. This is the basic workflow of the Smart Join blend-in PCR cloning kit. One step is to do purification of the PCR product, which is optional, but it can provide higher efficiency. And then you clone into the blend-in linear vector, transform the ligation product into bacteria, screen for positive colonies, and the positive colony formation rate is greater than 99%. It's highly efficient. And then do sequence verification. Okay, so the next step in the preparation that we recommend is to validate single guide RNA function in your cell line. Again, this is not essential, but it does help you help ensure the highest possibility of success. And the reason we recommend this is because not all single guide RNAs are created equal, and so it is best practice, our recommendation, to try multiple target sites and to pre-validate the best one. And to do this, you can use our indel check CRISPR indel detection system for validation. And so this system is highly useful for mainly for two functions. One is, as I'm talking about right now, CRISPR single-guided RNA functional validation, and also screening for positive clones. This is a three-component system. The first is the target site PCR kit, which allows you to, it provides a high-efficiency TAC DNA polymerase for amplifying your target region. The next component, the most critical component, is the T7 endonuclease 1 kit. And the third component is the blunt-end PCR cloning kit that I just talked about, the Smart Join kit. And one nice feature is that no genomic DNA isolation is required. You can just lyse your cells and then do the PCR and the T7 step in the lysate. So this is how the T7 endonuclease 1 assay works. It takes advantage of the low fidelity of non-homologous end joining because it uses an enzyme called T7 endonuclease 1, which detects mismatches that are generated as shown here. So if you do a transfection, you'll then get, by non-homologous end joining, you'll get a population of both cells with wild type alleles of the gene, as well as cells that contain indel mutations. Okay, and so when you create a PCR product using our PCR target site PCR kit, you'll then get a mixed population of fragments that contain either the wild type allele or with indel mutations. That mixed population is then heated to 95 degrees and then reannealed. That reannealing will lead to the formation of heteroduplex DNA, so that can be either one strand wild type and one strand mutant, or the two different strands will be mutant, but they'll have a different mutation. In both cases, the heteroduplex DNA will be digested or cut by this enzyme T7 endonuclease 1, which will lead to what's shown here on this gel on the right-hand side. Okay, the first lane 1 shows the PCR product that is undigested, and lane 2 shows the PCR product that has been after this melting and reannealing step and digestion with T7 endonuclease 1, we get two bands that shown by these asterisks of the predicted size, which tells us that we had a successful repair event. And so when you do this kind of test, you can validate the function of your single guide RNAs, because sometimes in some cases you will not see this at all, or you'll see some cases where some single guide RNAs give a stronger band formation than others. Okay, the next step we recommend in the workflow is to do copy number determination. Again, not essential, but this is important because for complete knockouts or mutagenesis, you might need to modify all alleles of your genome. Okay, now you may have noticed I mentioned all alleles and I didn't say both alleles, and that's because many cell lines are not diploid, especially cancer cell lines. Some cell lines, like HeLa, which is not a cancer cell line, but it's still a transformed line, have 3, 4, or even more copies of the genome. Now, you might expect multi-allele modification to be more difficult to achieve than single allele, but actually that's not the case. Double allele modification occurs more frequently than single allele. However, it's still good to know. It will still help you in your screening process to know how many alleles of your target gene you have. Now, if you know you have a diploid cell line, great, you don't need to do this step, but sometimes we've had customers say, well, I actually don't know how many alleles of copies of the gene are in this cell line. And so that's a project that I'm going to talk about next, where which to show you why it is important to determine the number of copies of your gene in your cell line. This is a project that we did at GeneCopia, a cell line engineering project we did at GeneCopia for a customer. And the project was to knock out the histone deacetylase 6 gene, or HDAC6 gene, in human MH7A cells. Now, the HDAC6 gene is located on the X chromosome, okay, and the MH7A cells are a human immortalized synovial fibroblast cell line. And so at the beginning of this project, we did ask the customer, do you know how many copies of this gene there are in the cell line? It was part of the initial consultation process. And the customer says, well, I actually don't know. Well, it's a good thing we asked this question because as you'll see momentarily. So we did a fish experiment using GeneCopia fish probes that recognize the X chromosome in this cell line, and that's shown here. So when we did that, we found that most of the cells contained 4 or 5 dots that lit up with this GeneCopia X chromosome centromere probe. Okay, and that was in comparison to the HT1080 cell, which is a cell line which is a known diploid cell line. In fact, it's a male cell line, so it only has 1 copy of the X chromosome, and it also has 1 copy of the Y chromosome, shown by these orange dots. The Y chromosome probe did not light up the MH7A cell line, so it's presumably female. Okay, so this suggested that this cell line, not only was it not diploid, but it actually had 5, it looked like it had 5 copies of the target gene, or at least the target chromosome. And so when we isolated clones, we found that indeed this cell line had 5 copies of the gene, because we were able to isolate a number of clones that had 5 different indel mutations in this gene compared with wild type. I'm showing 2 different clones here, and these are the sequencing results of each of these clones. And if you were to look, take the time to look carefully, you would find that there were 5 different alleles of this gene. So that's a great example of why doing a fish experiment first to determine gene copy number is a good idea. Okay, so if you want to know more about this project and how we did it, we have an application note on it. It's shown here. This is the download link, but again, as I did earlier, I will copy and paste this link into your chat box. And I just sent it to you via the chat box. If you don't see it, you may need to scroll down a little bit. Okay? All right, so moving right along, now I'm going to talk about screening steps, screening recommendations. So one of the most important screening steps we recommend in doing CRISPR cell line work is to isolate single clones post-transfection or transduction. Okay, so why should we do single clones? Okay, well, unlike RNAi, which actually most people use screen in pools for, CRISPR is usually used to make permanent changes to the genome. Okay, and so as I mentioned before, CRISPR gene modification can be very stochastic, even when you're using a donor, an HDR donor. So isolating single clones minimizes the potential effects of unwanted modifications resulting from random insertion, cell division, or off-targeting. So there are a number of different ways of doing single clone isolation. I'm not going to go into great detail about this. You may already know about them, but there are three major methods you can use. One is you can do the classical method of plating at limiting dilution and then picking single colonies off a dish using like cloning cylinders. A better method than that is to use fluorescent sorting if you have a fluorescent marker on a plasmid. Or if you're really unlucky and you're using a suspension cell line with no marker, you might need to do serial dilutions in multi-well plates, which does work, but it's very labor intensive. Okay, and the next thing to keep in mind when screening is to connect the genotype to the phenotype. And what I mean by that is we highly recommend that you screen the chromosome to make sure it has the genetic modification before you do your phenotypic screening. Now, you could use Western blotting, for example. Let's say you're doing a knockout and you have a good antibody. You could do Western blotting and look for clones that don't give Western expression. But it's also possible that you're just getting some gene expression, but it's below the detection limit. Instead, we recommend analyzing the genome and make sure it has the allele that you intended to have. Okay, so and to do that, again, that depends on the type of application you're doing. If you're doing non-homologous end joining, the procedure is different from if you're doing applications that require homologous recombination. By non-homologous end joining, we recommend, again, using the indel, the T7 endonuclease 1 assay, looking for indels that I mentioned before for validation. So we recommend isolating clones without selection 2 to 3 days post-transfection, generating PCR products using the primers flanking the double-strand break site, screening the PCR products using the T7 endonuclease 1 kit, or you can even do direct sequencing and then resequence positive clones by direct sequencing. If you're doing HDR-mediated applications, as I mentioned, the procedure is different. This shows what happens with homologous recombination. You get cutting by CRISPR, and then introduction of a donor leads to knock-in of whatever sequence is on the donor. This is a typical donor plasmid from GeneCopia. And so what we recommend doing is co-transfecting both the CRISPR plasmid containing the Cas9 nuclease and the single-guide RNA with the donor plasmid. In this case, you'll isolate about 2 to 3 days post-transfection. You'll apply selection for the donor cassette or begin to sort cells using the fluorescent reporter. Isolate single clones and then screen the single clones for correct integration by the PCR method known as junction PCR. Okay, so the donors, when they integrate, have two heterologous junctions, the 5' and 3' junction shown here. And so we recommend designing primers that flank both junctions. One primer comes from the unmodified genomic DNA sequence, and the other primer comes from sequences that are only encoded on the plasmid. And so when you do such junction PCR, you will only get amplification if you had integration of your donor at the correct site. And such an example is shown here. Lanes 1 and 2 are from a clone containing integration at the correct site, whereas lanes 3 and 4, this is an experiment we did. We isolated pure myosin-resistant clones, but they were negative for junction PCR, and so presumably these donors integrated into the genome randomly and not at the correct site. So again, you'll only get PCR products if the donor has integrated correctly. Okay, and finally, I want to talk about things to look out for, also known as pitfalls. The first is gene copy number. Again, I mentioned that. I don't need to go through this list again, but for complete you need to take into account the fact that some cell lines are not diploid. They may contain several more copies of the genome than you thought. Okay, the next thing to look out for is cleavage efficiency. So the efficiencies of indel formation by CRISPR can vary quite a bit, even with the best software design tools. People still see variations in CRISPR indel efficiencies ranging from 5 to 70 or even higher percent. And so this is a little bit simplistic, but um, in general, you might need to screen twice as many clones for a CRISPR guide RNA that has 40% cutting efficiency as you would for one with 80% cutting efficiency. So having a rough idea of the cleavage efficiency of your CRISPR guide RNA is useful. And so selecting for modifications using a donor might help. And then another thing to look out for is off-targeting. I didn't talk about this much, but you may know about this. It's even in the popular press about how CRISPR can lead to off-target modification, and that's because CRISPR guide RNAs can tolerate up to 5 mismatches between the target sequence and the chromosomal DNA. In addition, I did mention that CRISPR generally recognizes an NGG-PAM, but it can occasionally recognize a non-canonical NAG-PAM. And so all of these factors combined can lead to off-target cleavage and off-target modification. So this can be a problem. It depends on what you're doing, but off-target modifications can have adverse consequences on research results or patient safety. And in fact, a lot of journals now are requiring people to even do whole genome next-gen sequencing after a CRISPR experiment to make sure that no off-target modification occurred. So off-targeting should definitely be addressed. We do design CRISPR guide RNAs with the lowest possible predicted off-target potential. However, this is a prediction, not experimentally determined. Again, I mentioned that whole genome sequencing is usually recommended and required by some journals, but because it is the most comprehensive method and it's unbiased. However, it's very expensive. So if you had to, you can also use targeted NGS methods such as digenome-seq and vivo methods, which have been published in the past few years. Or you can sequence predicted off-target sites, which is an inexpensive and fast way to go, but it's heavily biased. But the bottom line is off-target modification of CRISPR should definitely be addressed. Okay, so with that, I'd like to summarize. I told you the workflow for CRISPR genome editing experiments must be carefully considered for aspects such as the type of modification you're using, the delivery method, the cell line you're using, etc. Other considerations must be taken into account for genome editing experiments such as transfection efficiency, copy number variation, and cleavage efficiency. And finally, I told you that GeneCopia offers many products and services, including plasmid design and construction, functional validation, validation and screening kits, and transgenic mice to meet your genome editing needs. All right, so with that, I'm going to stop and take questions. Okay, so the first question is, does your Cas9 protein have NLS? Yes, so NLS, if you don't know, for those who don't know, is nuclear localization signal, and the answer is yes. Our soon-to-be-released Cas9 nuclease does have an NLS on it. That's usually necessary for cell line-based work for delivering the Cas9 nuclease into the genome. If it did not have an NLS, it could still cut DNA, but that would be mostly useful for like in vitro validation experiments. Okay, next question. Can you send me your PowerPoint? Yeah, I can. We will make the slides available on the website within the next 24 hours, and also there will be an on-demand recording of this webinar, so you can view it again and again and again if you want to. Okay, so next question. What is the targeting efficiency or off-targeting effect when comparing shRNA or Cas9? That's a good question. I don't have any hard numbers for that. I I believe Cas9 is actually better off at off targeting, especially when you use the higher fidelity mutants, such as CRISPR Cas9, because those are the off targeting efficiency of the higher fidelity variants is actually near background levels. So those are definitely much lower than most shRNA experiments. But again, I don't have the hard numbers on me. Okay, so next question: can multiple genes be knocked out using the same construct? Yes, they can. You can. The sgRNA plasmids I showed before showed only one single guide RNA on them, but you can actually put two or more single guide RNAs on a single plasmid if you want to, and get multiple gene targets. Okay, next question. How much does it cost to make a Cas9 cell line? Um, Well, that really varies a lot on many factors. It varies based on the type of cell line it is, how difficult it is to work with, like its transfection efficiency, and also I couldn't tell you the price right now anyway because some of you are international customers, so you would need to go through a local distributor. But if you're really interested in finding out your specific situation, just contact me and I can help you out with that. Okay, next question. By CRISPR editing, genetic changes are inherited or not? They are in most cases they are inherited. In the case of the traditional Cas9 CRISPR Cas9 method, they are inherited because the DNA is actually permanently modified, so it will be passed down to the daughter cells. Okay, next question. How will that complicate the screening for all gene knockouts? So, if you're referring to multiple? No, you're referring to you're the person. This is the person who asked the multiple gene knockout question. It will actually not make it very much more complicated to do the screening for multiple knockouts versus single knockouts because it's all PCR based. So if you just use PCR products with primers that are targeted to the region of the gene, then you can just do multiple PCR products and multiple sequencing reactions. So it'll just be additional work, but it's completely doable. Okay, next question. If it's inherited, is it ethically acceptable? Well, ethics are a very good question. There's obviously been a lot of discussion about that recently, especially given the claim by the one group recently that created edited human babies. That's a very good question that I'm unfortunately not going to be able to address today. But it's really important, obviously, and needs to be worked out. Okay, so next question: do you sell knockout cells? By knockout cells, if you mean, do we have cell lines that already have genes knocked out in them? We have a very small number of cells right now that are pre-made. We have what we call cancer biomarker mutant cell lines. These are a small collection of HCT116 cell lines that contain either BRAF, EGFR, or KRAS mutations in them. And so, but if you're talking about also custom cell line services, we have that too. You would just need to contact us for a custom project. All right, so hey, these are all great questions, everybody. I really appreciate it. Are there any more questions before I go? Yes, someone just asked a question. Won't the immortalized cells lose the knockout with passage as they are prone to genomic rearrangements? That's a good question. Well, if you have a double allele knockout and the mutations are tightly linked, then you should not lose the modification unless it's really deleterious to the cell. If it's heterozygous, then yeah, there's a good chance you will lose it. And so it might be good to knock it in with a selectable marker. But that is a very good question. Okay, so next question. If I choose to do in vivo projects, should I take all those steps you shown here? The answer is no. Well, the answer is some of the steps, yes, but some no. In vivo work actually does require a different different type of screening workflow. But if you want to contact me, we can talk about that. Next question. Will the genetic information from the lentivirus or AAV get inherited? The answer with lentivirus is most likely yes, because lentivirus usually integrates in the genome. With AAV, the answer is most likely no, because AAV does not integrate, and so if the cell divides, the genetic information will probably get lost. It depends on what kind of modification you're doing. Actually, I correct myself. If it created a knockout, then it will get inherited. So I'm sorry. The answer in both cases, lentivirus or AAV, is usually yes. All right. So any more questions? Yes. Let's see. Does the virus the best strategy deliver in vivo? The best viral strategy in vivo is usually AAV, because it doesn't integrate. Although you can use lentivirus too. People do use that. And in fact, you can use integration deficient lentivirus. Okay, next, what MOI is ideal for lenti transduction in primary cells? That really depends on the cell type. The MOI is a good question. It is something to take into account, but we don't have much experience here with primary cells, so you would really have to determine this empirically. Okay, next question. Fibroblasts, for example. Unfortunately, I don't know the answer to that. I'm sorry. You'd probably still need to determine it empirically. All right, great. Well, with that, I'm going to stop. If you have any more questions, feel free to give us a call. Give me a call at 1-866-360-9531, extension 227, or send me an email, edavis@genecopia.com. And please feel free to visit us on the web at www.genecopia.com. Thanks, and have a great day.